the cell dry weight. So if you took cells, you dried them and got all the water out, the large majority after that would be proteins. But about 70% of the wet cell mass is going to be water, maybe even 90% in some cells. Proteins have a lot of different roles. They can catalyze reactions. They can serve as transport molecules. They help cells to move around. You already know most of these things. They provide framework for the cell. They help cells sense the outside. Remember those little bacteria you were looking at in the laboratory last week? They can actually tell what's going on around them. They can tell if a neutrophil is making their way up toward them to gobble them. They can sense the neutrophil coming because of the chemicals the neutrophil releases. And they do it through these protein receptors that are outside on the surface of the cell. Proteins are also involved in turning genes on and turning genes off. That's what gene regulation means. In Chapter 7, we're going to talk a good bit about how that all works, so keep that in mind. Proteins, you already know, are made up of amino acids, and we've talked about the 20 essential amino acids. Protein shape depends on, protein structure depends on shape, and the characteristics depend on shape, and that's determined by the amino acid sequence, the specific order of the amino acids, and we call that the primary structure. I think, did we talk about this? We talked about this a little bit. I feel like we have. But remember that amino acids can exist in the L stereoisomer, which is what we see being put into proteins. But bacteria actually use some of these guys, D stereoisomers, and they're just glued together differently. All of the same components are there. So you still have an alpha carbon, you've got that amino group, you've got the carboxyl group, and you've got a side chain, but they're just put together differently, and that makes them chemically different. And that's what Pasteur was looking at the beginning of this chapter when we looked at the historical glimpse. I feel like we talk about this a little bit too, and that we can group these guys. And if you look at the side chains of all of these, there are no charges there, and that makes them hydrophobic or nonpolar. And if you look at the side chains of this group right here, you begin to see some potential net negative charge typically because of all those oxygens that are out there on each one of those side chains, and in this case a nitrogen as well. And those potential partial charges make them polar, make them a little bit charged but not fully charged. So we actually call them uncharged because remember it's just that delta charge, a delta negative charge or a delta positive charge. But there is a little bit of electrophilic nature there because of that oxygen. Remember that? And that makes them polar side chains. And then these guys are also polar, but as you begin to look out, these guys are acidic. That means they want to, where those arrows went, they want to give off hydrogen. That's what acids do. They donate hydrogens. And these guys are basic, and you can see that because they've got side chains that want to draw hydrogen, and you can see that at each one of those positions where they're wanting to accept hydrogen, and so that makes those basic in nature. And that provides the characteristics of the polypeptide as it's being put together. If you put a hydrophobic amino acid, it wants to be down inside of that polypeptide chain. If you put a polar amino acid, it tends to want to be on the outside, out where the water is, and so it makes the protein fold in that way. The amino acids are held together in a polypeptide chain by what we call peptide bonds. Hopefully you remember this from anatomy and physiology. Those are covalent bonds, and they're a covalent bond between the carboxyl group of one amino acid, you can see that here, and the amino group of another amino acid. And these then are, again, kicked out via a dehydration synthesis. Water's kicked out, and you end up getting this carbon linked to this nitrogen to form this peptide bond right here. Those are covalent bonds, so those are reasonably strong bonds. It's a dehydration synthesis, and you can synthesize polypeptides that way. One term that students frequently get confused is they'll call a polypeptide a protein. Sometimes proteins are a single polypeptide, but many times biologically proteins are multiple polypeptides that have to come together. And even if you have a polypeptide, it's not necessarily a protein, even if it functions alone, unless it's folded correctly. Many times you'll find out that they have to be processed. They have to be folded in a certain way. Some things have to be added onto them to make them those polypeptides, those functional proteins. And so I'm sure you've talked a little bit about that in anatomy and physiology, and we'll look at it a little bit more here. But the ribosomes go about linking the amino acids together through this dehydration synthesis and forming the peptide bond that you can see right here. Okay, so the protein structure is key to function. The primary structure is just the order that we link the amino acids together. Then we've got secondary, tertiary, and quaternary structure. Some of you were having questions over, I'm going to call it winter break since we got a 3-day weekend last weekend, about LearnSmart. It wasn't working, so I actually jumped on and I did a whole bunch of it. It was kind of fun because I haven't done a lot since we were building it. But I got to these questions, and my brain wasn't all the way kicked in. I actually missed this one, so that's one of the ones that I missed. I called quaternary, tertiary, as I was trying to answer them as quickly as possible. I don't know if you figured it out yet. I don't encourage you to do this, but I was trying to take advantage of it. The faster you go, the more points you get for your correct answers. If you sit there for 5 minutes and then you get the correct answer, you get less points than if you answer it directly. But I'm grading you based on percent correct. So don't worry about getting too high in the ranking. Make sure you get as many of the answers as possible correct as you're going through it. Does that make sense? Okay. So I was going really quickly trying to see how high I could get my score up in a half an hour, and I missed a few like that little one. So if we look at a polypeptide and we start linking amino acids together, we can see this amino acid linked here, the amino group is missing from it, and then this amino acid is here, and then this one is here, and this one is here, and the carboxyl group would be over this way. And so we can start linking those together, and whatever this amino acid is followed by this one, that's going to be the primary structure. So the amino acid sequence. So usually you know that polypeptides, at least when they're being synthesized, start with methionine, right? You remember that from your other basic science classes. Start with methionine, and then the sequence on the gene, which gets put onto the messenger RNA, then dictates what the next amino acids will be. And so that's going to be that primary structure. And then depending what those side chains are, those R groups that are depicted there, that dictates how that protein is going to fold. Remember we just said those polar side chains are going to want to be outside, the ones that have some charge, and the non-polar ones or hydrophobic ones are going to be down on the inside. And we can start to see some structures that are built. One type of structure that we see is this, where the polypeptide folds like this, and it might fold like this and fold like this. And I was actually just talking about this to my immunology students yesterday, because immunoglobulins have a whole bunch of those beta sheets in long stretches, and they actually start to fold on themselves and make kind of a barrel structure. And so we call those beta barrels. But that's called a beta pleated sheet. When scientists first saw it and started to see the structure, they said, it looks kind of like the way we fold our bed sheets. And so that's how that name stuck. And that's one type of secondary structure. Another type of secondary structure is when we start to get helices, and I can't really draw it well, but you get a spiraling in the polypeptide chain, and then you can see it better if I erase that. You start getting cross-links or hydrogen bonding, ionic bonding between the side chains that are on those molecules, and it makes the polypeptide form into a helix. And sometimes we get helices, and sometimes we get beta pleated sheets. So if I take this arm and I make a pleated sheet, and then I take this arm and I make a alpha helix, which I can't do too well, but if I do that, I've got one type of secondary structure right here, right? And I've got another type of secondary structure right here. Everybody's with me, right? And then if these guys come together and start to interact, so the amino acids that are here and the amino acid side chains that are here start to interact, now we've got tertiary structure, right? Can I get you to fold with me? She's going to fold. So she might fold a little bit differently than me because she might have a different amino acid sequence. But if she starts to fold together and then we start to interact together some way, that's actually quaternary structure, right? Because now we've got two polypeptides that are coming together. Thank you. And so that's quaternary structure. That's this fourth level of structure. And most proteins end up having to have quaternary structures. Some protein complexes, we start to put complex if they get too big. Some protein complexes have 6, 7, 8, 9, 10, 13 different polypeptides that have to come together before they can perform the function that they need to perform. You know that a simple mutation, a change of one nucleotide in the DNA, could change any one of these amino acids. And if it does that, that can mess up this secondary structure, or this one, depending on where it is, which would mess up this tertiary structure and obviously mess up this quaternary structure. So that's protein structure. Besides folding, we end up getting domains, and one domain on a protein, a lot of proteins you have already learned about, at least in generic terms, use ATP because you learned a lot about metabolism. And so if those proteins use ATP, they oftentimes have a domain that we call the ATP binding cassette domain. So it's a domain that's specific to bind ATP, and it has to have a different structure. So when we look at new proteins and their amino acid sequence, a lot of times we can predict this protein must use ATP because it's got this domain that's involved in binding ATP. And there's a lot of other domains that we know the structure of, so we can begin to predict function of proteins because of that. You guys have done the experiment where you made egg drop soup. Have you made egg drop soup before? Started boiling water. You should do it sometime. It's just fun to watch because you're denaturing millions of proteins. Boil water, drop your egg right down in there, and all of a sudden the egg clear becomes egg white. The yolk starts to get hard. You're denaturing all the proteins down inside of that egg. We can do that process by just adding heat, which is what you do when you fry an egg or you make egg drop soup like I just described. Heat causes those alpha helices, those beta pleated sheets, and all the other secondary, tertiary, quaternary structure to unfold and fall apart. And that's what we say, we call it denaturation, when we heat proteins and cause them to unfold. When they unfold, they're no longer functional. And you might have noticed this arrow right here is a unidirectional arrow. That means it can't go back this way. Once a protein is denatured, it's very, 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 very difficult to get it to renature. And in the context of a cell, it's almost impossible. There's a whole system for clearing out those denatured proteins because it happens. Proteins are just little machines. They break just like all machines down inside of the cell. And there's a whole other system for clearing out those dead proteins that are down inside of the cell, chopping them up into amino acids, and then making amino acids so you can recycle them to make more proteins. So when denaturation happens, you get unfolding. We can do it by heat. We can do it by pH. If we go acidic or basic, you can get proteins to denature. And sometimes you can just add salts. If you go too acidic, you'll begin to break the bonds between those peptide bonds that we talked about. Carbohydrates are next. Hopefully you had some carbs for breakfast or somewhere along the way, because they're our energy source. They're our storage unit for energy, and you already know these two carbohydrates that are listed here. I'm thinking you've seen them in a lot of classes before. The top one is ribose. That's the carbohydrate that's going to be in RNA, and the bottom is deoxyribose. Can you see the difference between those two carbohydrates? There's only one very small difference between them. So the deoxy has lost that hydroxyl group. It's been replaced with a hydrogen. That's how it gets its name. It lost that oxygen right there, and so that becomes deoxyribose. Everything else about them structurally is identical. Those guys are obviously going to be down inside of DNA and RNA. We're going to look at some other carbohydrates here as well. But carbohydrates make up a good part of the structure of the cell, and they got their name because of this. Their name is carbohydrates. And hydrates means that water was added back to carbon. Did you know that? That's how they got their name. Beautiful. So the ratio is 1:1 always. You're awesome. I'm glad you knew that. But that's how they got the name. We add water, and you can see the formula for a carbohydrate there. We've got 1 carbon and 1 water right there on this, and then just depending on how big the N is, depends on how big that carbohydrate is going to be. So it literally means a hydrate of carbon. Here's a bunch of carbohydrates. You probably are familiar with some of these guys. You should get familiar with all of them. Carbohydrates usually exist in monosaccharides, but they can be linked together to make oligosaccharides or polysaccharides. Some that you know, deoxyribose and ribose, have 5 carbons. Others that you're a bit familiar with have 6 carbons. You can see the ribose here, and we always start counting carbon or carbohydrates from the oxygen. So there's the oxygen right there. So this is going to be the number 1 carbon, that's the number 2, that's the number 3, that's the number 4, and the number 5 is out here on this side chain. And sometimes we don't actually put the carbons on there. Sometimes we just draw it like this, and then we put a little bend out there. If we do that type of thing, then we'll put the hydroxyl out here usually, and the number 5 carbon is going to be right there at the kink. Students, I asked them a couple of points along the way last semester to count carbons in a couple of molecules we'll come to in another chapter or two, and they didn't realize that every time there was a kink like this, that indicated that there was a carbon in the molecule. So just want to make sure everybody's on the same page, so when I give you some homework down the road, you'll get the right answers. So, yeah, I know you love homework. So this is glucose. You've heard of it. You know glycolysis. You know how to oxidize glucose down to pyruvic acid or pyruvate, and then you know that it can go into the TCA cycle. That's that guy. Again, the number 6 carbon is out here on this side chain again, and so you can count around again from the oxygen that way. And then its cousin right here is mannose. Can you guys see the difference real quick between mannose and glucose? What's the difference? Describe it to me. Right here on the number 2 carbon, the hydroxyl group is on the opposite orientation on the plane of that molecule. And that's the only difference. Remember back to the historical glimpse, Pasteur was working with stereoisomers. That's what that is. And basically it functions differently, it tastes differently, and it does a lot of different things differently. Here's galactose down here. Again, if you look close, you can see that there's just a one change there. In this case, if we just compare glucose and galactose, it's right there on the number 4 carbon, and you can begin to see how we can make different carbohydrates that are very, very similar, except for just slight modifications. And here's fructose. You guys are hearing a lot about fructose in the media these days because many of us consume maybe too much of it in our soft drinks. So these are structural isomers or stereoisomers, and distinct properties, names. We get alpha beta forms depending on how that side chain is going together. And again, we can link them together. We can take carbohydrates and we can form them, link them together. This is one that probably most all of us had some of this morning because we use it as one of the prominent sweeteners in our diet. It's sucrose. It's the stuff that table sugar, the white granular sugar that you have on your table, is made of. And it's 1 alpha glucose and 1 fructose that are linked together. Notice again that it's a dehydration synthesis. We get water being kicked out, and so that sucrose is a disaccharide, and you're familiar with it. It's got fructose and glucose linked together. Lactose, that you've probably also heard of, that's the predominant sugar in milk, for example, and that's got glucose and galactose linked together. And then maltose is going to have 2 glucose molecules linked together. Link them together by dehydration synthesis between the hydroxyl groups, and then we can reverse that just like always via hydrolysis, and then water's kicked out and we get 2 monosaccharides back out. We can make long chains of carbohydrates, and one that you guys know of because many of you are writing on a large majority of it right now is this guy, cellulose. And cellulose is a major polysaccharide that plants make, and if you look at the bond there, the bond in cellulose, it's 2 glucose molecules that are linked together, and the bond is between the number 1 and the number 4 carbon right there. We don't make glucose, we make this molecule called glycogen. And that's how we store much of the excess glucose that comes into our system. If it can't be used to make ATP, then we and other animals will make some glycogen to store that. And look at this bond. It's also a 1,4 bond between those 2 glucose molecules. Our cells have plenty of enzymes that can break that 1,4 bond between those 2 glucose molecules and glycogen. But we can't break this bond. One student came to me after this lecture a while ago and said, we have a food shortage in the world. Why don't we engineer human cells to have the enzyme that can break those bonds in cellulose? Because then you could just chew on toothpicks all day long and you'd have all of the energy that you wanted if you could release the glucose out. And why don't we introduce that into humans? And my only response was, well, most of us already have too much energy storage in our bodies already, and we would be enormous if we could digest everything that we call fiber in our diet. If it was then digestible, which it would be, then we'd become enormous. It might be a problem down the road, and we actually use it for other things. If you've noticed your gasoline over the last 5 or more years, it's got 10% ethanol in it. And that ethanol is actually made from corn, and there are microorganisms that can easily break these bonds. And then when they release the glucose, it can be used to go into glycolysis, and then you can go on in and do fermentation with the pyruvate, and you can make that ethanol. So we have systems to do that. I just don't think it's a great idea to allow us as humans to get any more energy out of our diets than we already do. But you also know that we can feed some of our friends, those friends that go, those guys, you know them? Yeah, I'm pretty proud. One year I was up in Fort Worth during Fort Worth Dairy Days, and I won the mooing contest. So I'll have to give you some more later down the road. You might think I have mad cow disease, but I got this trophy milking stool that was painted like a Jersey cow, and I was pretty proud that day. But anyway, cattle, you know, they can eat grass, and they can eat all kinds of cellulose that's in that grass, and they can break it down, and they can get the energy out of it, and that's exactly what they do. But I just told you a lie. They can't do all of that. But the microorganisms that live in their gut have the enzymes to break those bonds in the cellulose. And they do it. They make other molecules, and then they feed those molecules to the cows so that they can get the energy out of that cellulose. And then there's this guy, dextran. There's some dextran out in the environment. We don't have enzymes to break it. Other fungi and others can break it. We use dextran for a lot of things because our body can't break it down, so we use it as a type of plastic, or at least a component of plastics, so that our body can't break it down. If we make shorter stretches, then we've got oligosaccharides. I used that term a little bit ago, but I want to make sure you got it. Oligo just means some. So you can make an oligonucleotide, and usually when we think of oligonucleotides, they're 10 to 25 nucleotides that are linked together, something like that. You can make oligosaccharides. Again, they're shorter chains of those monomers that are linked together. All of these guys have side chains. If you look at it, cellulose is unbranched. There's no branching that's going on the cellulose. Glycogen, however, has branches. So if you think about it intuitively, the main bonds are these 1,4 bonds, but when you go out and look at how the branch is attached, it's actually attached differently. Again, it takes a different enzyme to be able to cleave off that branch because each enzyme is specific for one specific chemical reaction. You guys know starch as well, and it's another polysaccharide of glucose that's glued together. Chitin's another one, and then agar also. Agar is what we put into that media that we play with in the laboratory. That was one of those important discoveries way back in Robert Koch's day. He didn't do it. Some other people figured it out, but to make media solid, they tried all kinds of things. You guys have made Jell-O before. Jell-O's pretty cool. You just take gelatin, which is a protein, you heat it up, and then you let it cool, and it starts to congeal, and it makes a semi-solid material. The more gelatin you put in, the more firm it gets. And so they tried that, and it worked kind of, but many bacteria secrete proteases into their environment. That's how they get their energy. They break down things in the environment. So as they were secreting those proteases, the solid media made out of gelatin, or Jell-O if you will, started breaking down because the amino acid, the peptide bonds in there were being cleaved. And then agar is a kind of polysaccharide that most organisms don't have enzymes which are capable of breaking it down. So it serves as a nice functional solid substrate. By the way, I told you guys I'm going to give you these notes, and I need to get lecture chapter 1 up there for you. If you don't see it in the next day or so, just shoot me an email and remind me that you haven't seen it yet, and I'll get it put up there for you. So you know about nucleic acids. They're used to put DNA together, use nucleotides to do that. And for DNA, we call it deoxyribonucleic acid for that reason that we just talked about, that the hydroxyl group is gone from that area right there of the molecule. And then RNA is going to use ribonucleic acids, or ribonucleotides to be strung together when transcription happens. And they're linked together via bonds between this phosphate and this number 3 carbon hydroxyl right here. And we'll look at that a bit here, and we'll come back and look at it a bit more in Chapter 7. So these are the nucleotides, and they include a number of different parts. We already said they include a carbohydrate, right? Either ribose or deoxyribose. They also include these molecules, depending on which type we're looking at, and we can break these up into 2 groups. We've got the purines over here, and we've got the pyrimidines over here. You can remember the purines because the people over in College Station like to make this joke that Aggies are pure, and so that will help you remember that AG are the 2 purines, and then the others are going to be the pyrimidines. I guess that's a joke. Anyway, so you also know that these guys, the purines, are obviously used in both DNA and RNA, and these 2 pyrimidines right here, the thymine and cytosine, are going to be used in synthesis of DNA, and these 2, these 2 are going to be used in the synthesis of RNA. Uracil replaces thymine and RNA. You should probably know that from anatomy and physiology as well. So we can start linking these guys together, and they're linked together by covalent bonds, and you can see that here. We've got this first nucleotide with its nucleobase here that happens to be a guanine, and it's going to be linked together by this phosphate to the number 3 carbon here, and the synthesis continues to happen that way. So the next phosphate that comes in would link right here, and then the next nucleotide would go down here at the bottom. And that's the synthesis process for our nucleotides. Again, we're making a polymer of nucleotides. You know, when you get DNA, in most cases, DNA is double-stranded. At least that's what we think of. But down the road, in Chapter 13, you're going to see that some viruses actually use DNA for their genome, and many times it's single-stranded DNA, not double-stranded DNA like you're familiar with. We'll talk about that when we get down the road. Likewise, some viruses have RNA as their genome, and many times the genomes are double-stranded RNA, which is a little bit different than what you're expecting to see too. But when nucleic acids become double-stranded, you can see the individual strands, and we usually think of the strands from the 5' end to the 3' end. So if we were going to read this sequence, we would read G, A, T, G, T as we go from 5' toward 3'. And if we read this one, we would read it A, C, A, T, C, again as we go 5' to 3'. So you have to think of the strands in those orientations. They have specific orientation. And then when they come together, if they have sequences that can base pair, remember GCs can base pair together and ATs can base pair together. And so if you get hydrogen bonding between those guys, then they'll begin to form double-stranded nucleic acids like you can see there. If you look down at the bottom of that picture here, they're beginning to show you, and you can kind of see, remember oxygen tends to have a net negative charge, right? And that amino group right there tends to have a net positive charge. So you get a hydrogen bond wanting to form there, and we're going to get one there, and we're going to get one right there. So the GC base pairs can have 3 hydrogen bonds. In the case of the AT base pairing, we can only get 2, and that happens here and happens here. Spatially, these guys aren't close enough together, so we can't get that bond, so we only get 2 bonds in the AT base pairing. So think about this. Remember the other day we were talking about water in liquid, solid, and gas form? Which one of those 3 forms has the most hydrogen bonds in it? The solid one, right? The solid has the most, and that's why we don't go play out in hailstorms. So think about this. Solid ice was going to break your head instead of your head breaking the hydrogen bonds, right? You're good with that. Because there are more hydrogen bonds. So if you have DNA that all is GC base paired together, or if you have DNA that's all AT base paired together, and assuming they're the same length, which one is going to be harder to separate those strands? GC. Why? There's 50% more hydrogen bonds. And just like going from liquid water to solid water, more hydrogen bonds makes it harder to break. So when we go from solid to liquid water, what do we have to do to go from solid to liquid water? Add energy, right? Usually we do that by raising the temperature. We can do exactly the same thing. We can add energy and break these hydrogen bonds that would be in between here. And we can get the two strands to separate. In this case, we call it denaturation. Again, the two strands come apart from each other, and we denature them. We can do that by adding energy. So it's going to take a lot more energy to denature this guy than it is to this guy. It's got 50% more hydrogen bonds over here, but when you add the amount of energy that's required cumulatively, it takes much more. Now, to go to something that's practical. We've got organisms. We talked about them in some of the labs. We'll talk about them more here. And some organisms like to live in ice. If you take them to room temperature, they die. Other organisms have to live up around 95 degrees centigrade where water boils. If you take them to room temperature, they may not die, but they won't be able to grow and replicate. So if you apply what you just learned here, what do you think the structure of one of those organisms that's called a hyperthermophile that has to live out at 95 degrees centigrade and divide its DNA at 95 degrees centigrade, what do you think its chromosome is going to have a lot of? They're going to be covalent bonds in the strands just like we saw here, but is it going to look more like this guy, or is it going to look more like this guy? Why more GC? Because that way the strands won't separate as easily, right? The more GC, the less likely the chromosomes are to denature. They also have some very stable proteins that help those strands stay together, but they tend to have higher GC content so that their DNA doesn't melt as easily. And those guys that live in ice tend to have lower GC content, verging on that AT content. Why would that be? What has to happen for DNA to replicate? Has to be unwound, and it also has to bubble. It has to separate for DNA to replicate. There are enzymes that come in, proteins that bind, that get the two strands to separate. And so in those temperatures, one has to happen, or in those two organisms, one has to happen in a very high temperature, one has to be possible in a very low temperature. We'll come back and we'll talk about that a good bit more down the road. Then there's lipids. Lipids are nonpolar. They're hydrophobic molecules. You guys played with these guys when you were making your pasta that time, when you pour the oil into the water. They have a lot of different properties. They're not soluble in water typically, at least not to a high degree, but they are very soluble in a number of different organic solvents, and that's because these guys are also nonpolar solvents. And that allows those organic nonpolar molecules to be able to dissolve down inside of them. We can make simple fats just by taking some glycerol, and I wish they had separated this. This is one fatty acid here, this is a second fatty acid here, and this is a third fatty acid here, and I wish they'd separated out those blocks. But again, we can do a dehydration synthesis and link these guys together and make this nice simple triglyceride. Do any of you have friends who make their own biodiesel? Have you heard of that? You have friends that does that? It's not hard. In the old days, like 10 years ago, McDonald's was actually paying people to haul off their used vegetable oil that they used to fry their French fries or their fish sandwiches or other things like that. Once you've used it so much it starts tasting burnt, can't cook with it anymore, and they had to pay somebody to haul it off. And so these clever people got this idea, hey, we'll haul it off for free if you'll let us haul it off. And they took it, and that used